Hi, this is Jose Luis de la Torre and welcome to my talk for the AAPA Meeting 2020. This is Brain-neurocranium mapping in a nutshell. I performed this research together with Christoph Solikoffer, Marcia Ponce de Leon and Bill Hopkins. Here you see an image of a human brain and an image of a chimpanzee brain. The most striking difference, of course, is that the human brain is much larger. But in most aspects, a human brain is simply a large primate brain. For example, with just a little bit of training, anyone can learn to identify patterns of cortical folding that are shared across species. These cortical foldings are called brain sulci, and some of them consistently coincide with the boundaries of functional regions in the brain. Now, this is not to say that the brain of our ancestors did not undergo substantial reorganization in at least a few regions. In fact, there are two regions where humans don't exhibit the great ape sulcal pattern and that are commonly considered hallmarks of human brain reorganization. The first region is the frontal operculum or Broca's area in humans. This area is highly involved in language production in our species and therefore thought to be of extreme importance during human evolution. This one also matches quite well the underlying situ architecture. The second region is the one around the boundary between the occipital and parietal lobes. This is clearly demarcated by the lunar sulcus in chimpanzees and other great apes and coincides with the boundaries of the primary visual cortex in those species. Humans don't have such a sulcus, or if we have it, is most of the time not present, and when present is in a much more posterior position. Well, can we know when and how changes in brain size and organization emerge in extinct hominin species? Now, for brain size it's quite simple because we can just cut a skull open, measure the volume of the endocrine cavity, and we will have a good estimate of brain size. Now, for brain organization, it is not that simple, but surprisingly, at least to me, it is still possible to a certain degree. If we create a cast from the endocranial cavity, like this, we will get an endocast from endocranial cast, which has surprisingly a lot of information. For instance, we will be able to see the position of cranial sutures, traces of vascular structures, and with some luck and in some individuals, so-called imprints which reflect the external morphology of the brain. Now, what happens in practice is when paleoanthropologists find a new fossilized skull, they will try to create such an endocast and examine its surface in search for so-called imprints that can tell us anything about the organization of the brain. So, how valid is this method? Can we trust all these findings? Here we see CT MRI data of the same subject. This is a human. We're going to compare so-called imprints and the actual brain sulci. So here we have a brain obtained from MRI, and here we see the endocast obtained from CT. Now, these images will go back and forth a few times, so please pay attention in the areas highlighted in red. As you see, there is a striking correspondence in a few regions, and this happens often enough in different specimens. So, endocasts do reflect the external morphology of the brain. Of course, this technique is still highly subjective and therefore susceptible to disagreements and controversies. Perhaps the most famous one is the position of the lunar sulcus in Australopithecines. This species had a relatively small brain size, just a bit larger than chimpanzees, and some authors, such as Ralph Holloway, claim that the lunar sulcus was located in a much more human-like position, while others, like Dean Falk, claim that it was in a much more ape-like position. In the last 2 decades, with the advent of biomedical imaging techniques and the widespread use of computational tools to analyze this data, some researchers have focused much more on analyzing the overall shape of the endocast rather than so-called imprints. Using these methods that can be more easily replicated by other researchers, we now know much more about the patterns of within and between taxon variation of endocrinal shape in humans, great apes, and even extinct hominins. And not only that, but we also know the same for brain asymmetry and even for postnatal brain development. What hasn't been done, however, is the actual quantification of the spatial relationship between the brain and the neurocranium, and especially of the patterns of covariation that operate between the two structures. And that's the focus of this study. By now we know that modern humans have a very unique neurocranial shape. Both the external surface and the internal surface of the neurocranium are very globular. This is not even seen in other members of the genus Homo, not even in the large brain Neanderthals. We also know that modern humans have a very large parietal bone in proportional terms. And we're starting to know when these patterns started to emerge in time. All these are landmark studies. They have been replicated, and by now we can consider perhaps these results facts. What we don't really know is what to do with these facts. What does it entail to have a globular brain case or a relatively large parietal bone? For instance, can we know anything about the brain organization of the Neanderthals who also had equally large brains but less globular ones? Here we use MRI data and in some cases also CT data of the same subject of humans and great apes. We first extract features of interest on the external surface of the neurocranium. We do the same for the endocast and for the brain and extract the overall shape of the endocast. This is what a complete dataset looks like. We did this in 41 humans and 24 chimpanzees and also in 2 gorillas who were used for outgroup comparisons. We then computed the average configuration of the human brain. We did the same for the human neurocranium, the chimpanzee brain, and the chimpanzee neurocranium. We also visualized the two structures together to examine topographical relationships in each species. Finally, we compared the average human configuration with that of chimpanzees. We did this in a species-independent space. We did this for the brain, for the neurocranium, and for both structures. In all these comparisons, the red ends represent the chimpanzee configuration and the blue ends represent the human configuration. Okay, let's look at these comparisons in detail. This is the difference between the human and chimpanzee average neurocrania. The first thing we see is that the human brain case is much more globular, especially the endocranial cavity. Second, we see that humans have relatively larger parietal bones. And third, we see what we know since more than a century, that there was an anterior displacement of the foramen magnum, reflecting that we are obligate bipedals. So, what about the brain? To our knowledge, this is the first quantitative comparison of the relative position of homologous brain sulci in humans and chimpanzees. We also mapped in this species-independent space the position of brain sulci that are specific to humans or to great apes. We also found that humans do have more globular brains, but the magnitude of this difference is much smaller than the one we found in endocasts. More importantly, we found that the region with the largest differences was the opercular region, and especially the frontal operculum, which corresponds to Broca's area in humans. So, what can we say about the parietal lobe? Well, since humans don't have a clear landmark on the dorsolateral surface of the brain, it's difficult to say anything. So this can only be resolved by looking at the underlying CT architecture. What we can say, however, is that even though humans appear to have larger parietal lobes than chimpanzees, the magnitude of this difference is much smaller than what we were expecting initially. And this difference appears to be even smaller when we focus on the superior parietal lobule that we can see here from the top of the brain, or when we focus only on the medial plane. Okay, all this was about mean configurations. So what about patterns of spatial variation? We ran two separate principal component analyses. one focusing on brain morphology and the other one on neurocranial morphology. In both of them, only one dominant component was found, PC1. In other words, principal component number 2 was already very small. Naturally, PC1 already separated the two species, and when gorillas were projected onto these spaces, they lied in both cases in the chimpanzee cloud. We then ran a third analysis, this time using both structures together, and we found the same results. What we didn't find was an emergent pattern, let's say in PC2 or PC3, accounting for cooperation between brain and neurocranial features. And we can see all this in physical space. So these are the mean shapes. These are the differences between the human and the chimpanzee mean configurations. Now, these are the patterns of variation along PC1. As you can see, these images are almost the same as the one on the left, meaning that the mean shapes are more or less 2 standard deviations apart along PC1, and 2 standard deviations along PC2 represent so little variation that it's even difficult to see it here. Again, when we run a principal component analysis of brain features together with neurocranial features, we see that the patterns of brain variation pretty much stay the same, suggesting that the influence between the two structures is minimal. And we corroborated this by using 3 separate methods, all of them providing evidence for independent brain and neurocranial reorganization during hominin evolution, the title of our paper, which was published at the end of October last year. To conclude, the reorganization that we see in the brains and the neurocrania of our ancestors did not evolve in concert. These changes might have occurred in parallel, but they were not related. A logical and practical implication of all this is that the overall shape, relative size, and position of the neurocranial bones should not be used to infer anything about the organization of the brain. Also, another important implication of all this is that the examination of so-called imprints remains really important. Another thing is, one of the regions that underwent the largest amount of change during this time might have been the opercular region, in particular Broca's area. And finally, the changes that we see in the neurocranium might obey, in humans, might reflect just increased encephalization and obligate bipedalism, in particular, an anterior displacement of the foramen magnum. So, thanks a lot for watching. I also would like to thank the funding agencies, the Swiss National Science Foundation and the Schultz Foundation. I also want to say thanks to my 3 co-authors and every member of the Computational Palenthropology Lab and the Anthropological Institute and Museum in Zurich. I want to say thanks to the entire city of Zurich, and now I'm not there anymore. I'm right now a postdoc at the Fetal Neonatal Neuroimaging Center of Boston Children's Hospital and Harvard Medical School. And you can find here my contact details. I will be very happy to hear from you. Thanks again.